So maybe, Dr. Sands, there's another question here. It says, do we know why different fusions have such different PFS on selective TKIs? For example, 36 months for ALK versus 17 months for RET. What can we do against this?
Yeah, so keep in mind that the initial drug for ALK was actually closer to the 17. It was actually less. And it's really the newer drug now for ALK that's at 36. So the specifics in the answer to this question really has to do with not only the fusion, or we could also even say mutations, but so just to make it more broad, the genomic alterations, how effectively we're able to target that and hit that, and even just the effectiveness of the drug can come from some of the specifics in how effectively we're able to target these cells. But on top of that, then, is potential resistance mutations or alterations that develop. And so a drug may be very, very effective, but ends up having common resistance mutations that show up. So if we talk about this in EGFR mutations, so the first one that, as I outlined at the beginning of my talk, so erlotinib and gefitinib, those are the first generation EGFR TKIs, so targeting these sensitive EGFR mutations. We would commonly see a mutation called T790M show up, and that would then be a resistance mutation. Well, then the drug osimertinib developed, and that was a way of targeting T790M, but it also kind of more broadly hit a lot of the other targets that these first generation EGFR TKIs hit as well. Not all of them, but a lot of them. And so now osimertinib is a drug where we're seeing a longer disease control than we did on the initial drugs. And so we're seeing it both in mutations and in fusions where we're developing more effective therapies that work for longer. Are there inherent differences with ALK and with RET where RET is something that we'll never be able to target as effectively as we have ALK? I mean, RET is much newer. So we have the drugs are newer. The drugs, as Dr. Gaynor outlined, are pretty specific to RET. But is there a way that we can then improve upon that? And what is the resistance pattern that develops? And then looking at that resistance pattern to try to find ways of preventing those mechanisms of resistance. This is a process going forward, and I'm very confident that further down the line we'll find better and better therapies for RET as well. But these initial ones are an excellent start, as they're very effective and also well tolerated.
Yeah, I would echo that. You know, I would think of a question like this in three different categories, much like you did, which is there can be differences in just the fundamental biology of the driver, that is, ALK versus RET. So that's one. Like the cancer cell may care more about one, or one can be a more aggressive driver. That is, it sends signals that are just more powerful to the cell, or it leads to an earlier, a greater ability to metastasize. So there can be differences in that. It's hard to study that in the lab, but there can be biological differences. As Dr. Sands was alluding to, the second big thing is there can be drug differences. These are really the ALK. There are successive generations of targeted therapies, whereas these are the first approved RET inhibitors. But then the third thing that I would caution against is the statistical aspect, which is that when we come up, when we see these medians, if you look into it, these are probabilistic models based upon how long patients have been on the study and what's happened to the patients who are currently in the study. The Rett trials have only been open for a little over 3 years. So it's hard to reach an average of 3 years if they haven't been open that long. Most patients who have enrolled on the Rett drugs have really enrolled in the last year or two. So some of it also has to do with length of follow-up on the studies, because I think the point is that drug development has changed a lot. ALK was discovered 2007, whereas and we had the ALK inhibitors in clinic first approved 2011. We've had a lot more follow-up on those, whereas these novel RET inhibitors really have only been in the clinic for 3 years. So that's another important part. So we definitely want to see what happens long term with those drugs.
Yeah, that's an important point. That the RET inhibitors in many this wasn't all first line, and so when we're talking about ALK, we're talking about first line as well.